inference method like best fit, reveal, BIBN, Bayesian inference approach for a Boolean network. Actually, they limited the regulatory genes only by 1 to 3. So they have the scalability, they are not scalable. We like to develop we would like to develop a model that are scalable. Sorry, I cannot move slides.
So what's the problem, Shohag? You cannot move the slides, or?
I cannot, yes, I cannot move my slides.
Okay, I'll try to contact. So is it in your computer or it's something that is centralized in the platform?
Maybe it doesn't happen in my computer.
Okay, so that's it. I would suggest that you start them over.
Okay, now I can move my slides. Actually, there are also other methods, mutual information methods. They are actually the limitation of the methods. They cannot infer regulatory genes, and next slide, please. Yes, I'd like to define dynamic accuracy. Let's first define the genoid dynamic consistency. Genoid dynamic consistency is the similarity between the observed gene expression data, for example, here B1T, and the estimated gene expression data B prime 1, this column. Okay. And here comparison can start from t is equal to tau plus 1 because tau is the maximum time step lag. So now I'd like to define dynamic. So dynamic accuracy is just the average of genoid dynamic consistency over all genes. And then structural when the gold standard network structure are available, then we can compare the structural accuracy between the target network and inferred Boolean network. In our study, there are 3 important metrics are considered for performance analysis, precision, recall, and structural accuracy. This is our overall framework of NNBR. Real value gene expression data is converted by K-means algorithm. Then binary gene expression data is taken as an output for MIBNI. I would like to note here there are 2 stages in our algorithm. First stage we will use MIBNI. It is our existing algorithm because it is already proven that it can infer small scale with very good result. That's why we applied MIBNI in the first step. If it finds a good optimal solution, then regulatory genes here. Otherwise, a GA with neural network are applied to find the optimal solution here. Okay, then finally all the results are constructed. All the results are constructed Boolean network. Okay. Then the framework of our genetic algorithm. Genetic algorithm is initialized a set of random solution which are called populations. Then two parent solution are selected by Rowlett-Hill operator. Then we have used a crossover mutation operator to generate offspring. Then replacement is occurred when offspring replaces parent if offspring is better, is superior than parent. And this algorithm is continuous until a stopping criteria is met. Here is our chromosome representation EGA. There are two main constraints for GA representation. First constraint Kmax, it doesn't allow not too many regulatory genes, and a regulatory gene with lower mutual information with the target gene, then it is discarded from this chromosome representation. So here chromosome is represented by binary vector plane. Here ith value of represented by the 0 and 1. 1 means regulatory genes is included in the chromosome. 0 means excluded from the chromosome. This is our chromosome representation. Then we can evaluate chromosomes from here, this example. Yeah, we assume that the maximum time lag tau is 3 is set in our study. So 9 input neurons and 1 output neurons are generated. The neural network learns Boolean gene expression data using back propagation algorithm, which is a gradient descent method to minimize mean square error between the estimated output and actual output. Then we can define fitness, which is 1 by 1 minus this equation, where here gamma is a white factor and k is the number of regulatory genes, and c b prime is a dynamic consistency. Here the better solution means the higher fitness. Then we have used selection Rowlett-Hill selection operator to choose two parent solution from the populations, and the selection probability of a chromosome is proportional to the adjusted fitness, which is equal to A into fitness plus B, where here H is the adjustable parameter that selects the probability of a best chromosome to H times of that worst chromosome. Fmax and Fmin are the maximum fitness values and minimum fitness values in all the populations. Actually, it helps us to avoid premature convergence in the genetic algorithm. Then maximum studies used 1-point crossover, 2-point crossover operator. Here we devise a new crossover operator. If the bit value between two parents are same, then directly copies to offspring. Otherwise, a bit value between two parents are selected at uniformly random. Then we have used mutation operator. Uniform mutation operator. It just flip the bit value between 1 to 0 and 0 to 1, and we have applied mutation probability with very small probability 0.01. And replacement and stopping criterion, replace offspring when offspring is superior than parent. And we stops the GA after 1000 generations. And there are two types network are used in our study. First on artificial network and second on real network. This is the experimental setup for artificial network. We randomly structure artificial network, which are generated by Barber-Shard blood model, and 300 BA random networks with different network size, such as B is equal to 10 to 100 network size, and interaction are 2.5 multiplied with number of nodes. And maximum time step was set up B plus 20. And performance analysis, structural analysis, we have analyzed structural accuracy analysis, dynamic accuracy analysis, and running time for the artificial neural network. And here is the structural accuracy comparison. So we classified all the target genes according to the number of incoming links 1 to 10. So if you see here, when the number of incoming links increases, the accuracy decreases in all parameters, precision, recall, and structural accuracy. However, our method is achieved higher structural accuracy. Which means that our method is robust than other methods. We have also computed dynamic accuracy here. If you see that network size increases, first figure, network size increases, the dynamic accuracy decreases. But NNBI and GAPNI always found the dynamic accuracy is 1. So which means that these two algorithms always found optimal solution. And this is not surprising because in our first stage, we have applied MIBNI, our previous algorithm. That's why, this can make a confusion because in the previous slides, structure accuracy is for NNBI is greater than GAPNI. In an effort to discover this reason, we have compared the convergence speed between NNBI and GAPNI. And if you see in the second figure, the GAPNI is faster, where NNBI is slower, because GAPNI uses incomplete truth, Boolean truth table, where NNBI is uses a complete Boolean truth table. And we also compared. Yes. We also computed running time for for all the methods. Our method was slowest because of the generative search, where others uses others methods are fastest. However, our accuracy is best than other methods. And we have used here the small scale network results. We have used 4 networks here. ECL cycle net, it has 2 types of data set, which is called CDC15 and CDC28. And in Silicon and Silicon 2, these are benchmark data set from Dream 4 challenge, Dream challenge 4, Dream 4 challenge, then SCBC network. And large scale network, our method is applicable to large scale network or not. So we have generated 5 types of data set using Gian NetWeaver tool. Then we have, this is the result of small scale networks for dynamic accuracy and the structured accuracy. In both cases, our method performs well than other methods. And also as well as large scale real networks. For both cases, structural accuracy and dynamic accuracy, our method outperforms than other method. We also.
You should really wrap up.
We also analyzed our structural accuracy against noisy gene expression. Noise is created by flipping the Boolean value at 5% probability and 10% probability. And both cases, our method outperform all the other methods, which means that our method is robust, is also in the noisy gene expression. And here is the summary and contribution of this work. NNBI overcome the limitation of the scalability problem. Okay, thank you. NNBI, here's a summary for our works. NNBI overcome the limitation of the scalability problem and regulatory rules or functions. Actually, it's a 2-stage algorithm. If MIBNI fails, then NNBI is applied to find optimal solution. And chromosome representation constraint and crossover for an efficient search in our framework was implemented. A neural network replace and update rule table for regulatory function. representation, and extended time lag parameter between the regulatory genes and the target genes in NN. NNBA significantly outperform and the other method for larger scale inference problem in terms of both structural and dynamic accuracy. Future works, actually our NNBA method is slowest than other methods. So we will use parallel implementation for NNBA in the future. And we'd like to conduct, we would like to combine steady state gene expression data with time series gene expression for better structural performance, especially in the noisy gene expression data set. And we hope we will develop new approach to infer genome scale network. Until now, we have just infer 500 gene network. So in future, we'll develop a new method that can infer whole genome. So thank you for your attention. Please, if you have any question, then you can ask me.
Okay, thank you very much, Shohag. So unfortunately, we don't have time for questions. I know this is a bit cold, but I mean, if this was live, you would get a round of applause here. Unfortunately, it's not possible to do it virtually. And we should jump directly to the second presenter of the session, who is Taiki Fuji from Hitachi Ltd. And we're going to hear about feasible metabolic pathway exploration technique using chemical latent space. I would like to remind the attendants that they can ask questions through the panel that you have in the web, right? And then I can pass them on to the speakers. So thank you very much. And Taiki, floor is yours.
Sorry, I'm Taiki Fuji from R&D group of Hitachi. Today, I introduce this title, Physiometric Pathway Explanation Technique using Chemical Latent Space. First, I'd like to describe the background. Microbial production of chemical compound is an important contributor to promote sustainable industries. To design process for biosynthesis of used target compound, design of target compound and host, metabolic pathway design, and DNA sequence design are performed. Especially in metabolic pathway design, physiometabolic pathway to the target compound is required. To explore physio pathways, not only examine a combination of well-known enzymatic reactions registered in public database, but also finding non-registered potential enzymatic reactions that can catalyze the desired structure change are necessary. To achieve this, what are required to explore physiometric pathways? There are three major technical challenges. The first one is how to computationally represent chemical and enzymatic reactions without missing structure information. The second one is how to design physical pathways by combining a huge number of potential enzymatic reactions. And the last one is how to evaluate the relevance of the physical pathways. For the first challenges, chemical encoding and reaction representation techniques should keep the overall backbone structure involved in the substrate specificity of enzyme reactions. Also, it requires decodable numerical expression to chemical structure and manually predefined reaction rule-free representation. For the second challenge, exploring combination reaction within reasonable time is required. And first, for the third challenge, feasibility check and pathway scoring are required. To solve challenges, we propose three-step explanation technique. This is reaction feature calculation, pathway design, and pathway scoring. Specifically, first step, representation of chemical and reaction using chemical latent space. Second step, binary differential evaluation-based combinatorial optimization of reaction in combination with subset method. Third step, neural network-based reaction possibility prediction method for pathway feasibility values. For representation of chemicals, we use a data-driven and structure-based representation. Actually, we utilize a chemical latent space obtained from variation autoencoder VA method, which is one of deep latent modeling technique. The VA technique we used has a junction tree decomposition. The tree decomposition is effective for not only evaluating the molecular similarity of structure between small compounds, but also representing the overall backbone structure of compounds. The right figure is example visualization of two-dimensional chemical latent space. In the chemical latent space, the chemical structure are represented in continuous space. As shown in figure, a structure change can be expressed by adding or subtracting the vector in latent space. Thus, a variety of potential reaction and intermediate compound can be easily created and explored using computational algorithms. By using chemical latent space, we represent enzymatic reaction without manually predefined reaction rules. A reaction feature vector is generated by subtracting the latent vector of the main substrate from that of main product with the following equation. For example, the reaction feature vector of EC2711 subtracts the hydroxy group and adds the phosphate group to alpha-D glucose. So the right figure shows the example of enzymatic reaction using chemical latent space about EC2711. By decoding a latent vector of product obtained by adding the reaction feature vector to the latent vector of substrate, we can obtain the structure of the product in which the desired structure change occurred. In this manner, all reactions of metabolic pathways are encoded to reaction feature vector and stored in the reaction feature DB. Sorry. Next, we explain the pathway design and candidate pathways. To explore a combination reaction within reasonable time, we propose to use differential evaluation-based combinatorial optimization with subset method. We address the pathway design as an optimization problem that minimizes the squared error between the pathway feature vector and sum of reaction feature vector in set. The pathway feature vector is computed as difference vector obtained by the latent vector of the target and start compounds. The minimization problem is dealt with the nonlinear integer program NLIP problem. So since it is difficult to apply the optimization method using differentiation to NLIP problem, binary differential evaluation algorithm is utilized for solving the problem. To reduce the calculation amount and enhance search efficiency, M-size reaction feature vectors are randomly selected from the reaction feature DB and built as a reaction subset. All candidate pathways are then constructed by ordering all combination of the reaction feature vector in the set. Simultaneously, intermediate compound in the candidate pathways are reconstructed using decoder of the VA. Finally, we remove the unrealistic pathways by molecular weight check. We also developed pathway scoring method to evaluate the pathway feasibilities. We developed ensemble neural network-based reaction possibility prediction method to validate the relevance of reactions. The reaction possibility prediction is carried out using voting scheme that averages the output of sets of discriminators trained with different data sets. The voting scheme is an effective method in terms of reducing rejection rate and improving the accuracy rate. In the training, the level of reaction registered in public DB is set as real 1, otherwise virtual 0. Virtual means that reaction is virtually calculated on computer and not registered in public database. Finally, pathway feasibility value is obtained by multiplying these reaction possibility values. These are data sets for VA training and exploring pathways. The V training dataset of SMART consists of zinc dataset used in the V paper and compound data acquired from cake compound. Over 260,000 pieces of compound data were prepared under this condition. The enzymatic reaction dataset for pathway design consists of about 10,000 pieces of reaction data acquired from cake pathway and 300,000 pieces of virtual reaction data for training reaction possibility prediction. To verify performance, we apply the proposed technique to non-registered pathways. This figure shows the two enzyme reactions of validation pathways that are non-registered pathways for producing the target compound 2-butanol. For validation pathway A, we apply the proposed technique to pathway when 2-butanol was set as target compound and 3-oxypentanate as a precursor was set as start compound. For validation pathway B, pathway examination was conducted with proposed technique using acetylene, which is precursor as a start compound. This pathway includes non-registered reaction from which 2-butanol is produced from 2,3-butanol. As a result, we explored Fiji pathways connecting the metabolic pathways. We confirmed that each reaction feature vector generated by substrate and product described in each study was very similar to reaction feature vector of the EC number described in those studies. So validation pathway A was reported by reference 3 and EC number of the corresponding reaction is 4114. The reaction of EC 4114 registered in KEGU is a reaction from which acetone is produced from acetoacetate. And validation pathway B was reported by reference 4 and EC number of the corresponding reaction is 42128. The reaction of 42128 registered in KEGU is a reaction from which propanol is produced from propan 1,2-diol. And the left figure shows the transition of the number of candidate pathways when the number of reputation was set to 2,000 and the subset size was changed from 100 to 1,000 in step of 100. The reduction rate tends to slow at subset size around from 400 to 500. And right figure shows the result of measuring time per repeat on the PC having the computer specification shown in the table. From the result, when the subset size was 500, the explanation was performed in about 10 seconds per repeat. These are limitations of our technique. Available compound are short chain compound and their SMART not including dot and asterisk. That means ionic bond and wildcard. And it is necessary to set short pathway due to using subset method. Here's a summary of my talk. Our proposed technique consists of 3 steps for candidate pathway explanation. As a result, we found Fiji metabolic pathways, including 1 or 2-step non-registered enzymatic reactions. Regarding future challenge for chemical VAE, we need to improve the compound reconstruction accuracy and dealing with the compound excluded in this research. And we will also improve the proposed technique so that multi-step pathway can be explored more accurately. And this research is based on research obtained from project commissioned by the New Energy and Industry Technology Development Organization. And we would like to thank Professor Michiro for his valuable comments. Thank you for your kind attention.
So thank you very much. Thanks to you, Taiki, for the very nice presentation and for keeping it in time. So we have room for 2 or 3 questions. So I will read them loud. So the first one comes from Dick de Ride, and he says, interesting approach for the reactions with multiple substrates or products. Do you create 2 reaction vectors or somehow decide on the main one?
Oh, sorry. Okay, main one. Okay.
Okay, so there's only 1 vector per reaction.
Can I might show you my slide. So the main and main substrate and main product is defined as different based on the KEGG pathways. KEGG pathways is okay. Thank you. Okay, sorry. KEGG pathway is a main reaction in main product and substrate is displayed in the base pathway. So by using KEGG pathway, we define the main product and main substrate. Okay.
Okay, we have another question from Alt Jan van Dijk. So, would it be relevant and would it be possible to include experimental conditions, for example, the pH?
Oh, sorry. What do you mean?
So I guess he means to constrain the models, right? So when you try from the latent space, when you try to decode the suggested molecule, to constrain it somehow depending on experimental conditions such as the pH, so that the molecule can or cannot be protonated and things like that. So this is my interpretation. So using experimental values as a constraint for the decoding process of the model.
Yes. I think it's a perfect method. It doesn't perfect method. But based on the real data of registering KEGG pathway, so by using a real data set, we meet the relevance of the, yes, I suppose real data, yeah.
Okay, so I also have a question of my own. So for training the autoencoder, you use a combination of the zinc and the KEGG compounds, but do you constrain somehow the zinc compounds that get into the training? Because otherwise, I mean, I presume that the molecules that you generate might be quite different from the ones that, from metabolites, basically, because zinc database contain any type of chemical structures.
In the training, in the VA training.
So you're showing the autoencoder, you're showing it the 260K molecules that, as I see here, they come from zinc and from KEGG. Right, so KEGG are obviously metabolites, but the zinc database might contain any type of small molecule.
Yes,
which means that then when you decode this from the latent space, depending on the balance of this training, the molecules that you get might not resemble at all metabolites. How often does this happen?
Okay. So yes, his question is right. So our technique can decode it like a long chain compound and microcycles. Yes. So in the JTV paper proposed to use a zinc dataset, and the accuracy rate is at 70%. But by using KEGG compound dataset, the accuracy is reduced to 56%. So it's difficult to measure pathway compound. So, but we need to improve the VA technique. Yes.
Okay, thank you very much. Then we should move on to the next speaker, who is Yahui Long from the Nanyang Technological University in China, and he's going to tell us about ensembling graph attention networks for human micro-drug association prediction. So Yahui, the floor is yours.
Okay. Hello everyone, my name is Yahui Long. I'm a joint candidate PhD from Hunan University of China, Nanyang Technological University, and the A-Star Research of Singapore. Today, my topic is assembling graph attention networks for human micro-drug association predictions. Sorry. This is outline. I'm going to introduce research backgrounds and then present the details of our proposed method. Following that, I will share the experimental results and the analysis. Finally, I will make a conclusion for our work. Micro is very important to human health. For example, it can protect against the pathogens, generate essential vitamins, facilitate the medicine precision, and so on. However, the imbalance of micro-communicatives can also lead to various human infection diseases, such as obesity, IBD, liver disease, and even cancer. Therefore, micro is considered as a new therapy target for medicine, for precision medicine. Nowadays, the development of new drugs is greatly slow. Moreover, the problem of drug resistance is increasingly serious. So there is an urgent need to develop effective method to predict micro-drug association. In silico method of identifying micro-drug association is an alternative strategy. There are many advantages to predict the micro-drug association, such as assisting the screening of compound candidates for drug discovery and facilitating precision medicine. However, it is challenging to predict the micro-drug association. On the one hand, there are large amounts of prior biological knowledge available, such as micro-drug associations, drug chemical structure information, and microbial genome sequence. On the other hand, in the network, it is common to have some isolated nodes, such as N1 and N2. We name such nodes as new nodes. They have no links connecting them to neighbors. As we have no training dates for new nodes, it is such very challenging for computational model to predict the associations. In this work, we propose a novel computational framework to predict the micro-drug association named ECCB. In particular, we use graph attention network to deal with this important problem. Here is the flowchart of ECCB. First, we can construct features and graphs from different databases. And then we design a two-layer model with node level attention and graph level attention to learn node representations. With the representation for nodes, we can use them to calculate association probabilities between microns and drugs. In this book, we use different biological data to construct rich features for microns and drugs. For microns, we first download microgenome sequence from NCBI database, and then use one-hot encoding to encode the genome sequence. As a result, we can obtain original feature matrix. To extract more useful information, we further use PCA on the original future matrix and finally obtain microfutures. Similarly, for drugs, we first download drugs chemical structure information from drug bank chemical drug bank database, and then use structure information to measure the similarities between drugs. Finally, we generate the drug futures from the drug structure similarity matrix. To predict the molecular drug association, we construct three different types of biology networks. They include a molecular drug bipartite network, a molecular drug heterogeneous network, and a molecular disease drug heterogeneous network. As the molecular drug bipartite network is an actually existing association network, we can naturally use it to construct a real molecular drug network named Net1. Net2 and Net3 are two networks that are predicted by metapasses from the two heterogeneous networks respectively. So there are actually virtual networks. This table summarizes the statistics for each molecular drug network. After deriving the features and the graphs, we can use them to learn representations for molecules and drugs. As we all know, graph neural network has powerful capability of drawing its graph structure dates. Therefore, in this book, we use graph neural network to learn node representation. Here is our overall architecture of representation learning. We first take a micro-drug network and node features as inputs. And then we use a GCN layer. And then we design a hierarchical attention mechanism to learn node representation, namely node level attention and graph level attention. Node level attention aims to capture the importance of node-specific neighbors, while graph level attention is designed to capture the importance of different graphs. Next, I'm going to introduce node level attention and graph level attention in detail. First, we introduce node level attention. Here is the procedure of node level attention. The inputs include a micro-drug network and node features. We first use a GCA layer to learn original node representation, and then use a GAT layer to further update node representation. As a result, we can obtain a representation for each node in the network. For example, given a node N1, we first use GCN to capture the features of its global neighbors such as N2 and N3. Following the GCN layer, we use GAT to present the features of its local neighbor, such as N4 and N5. It should be noted that GCN considers the importance of neighbors equally, while GAT considers the importance of neighbors differently. Each node in different graphs may include diverse semantic information. For example, we can derive different importance attention scores, beta 1, beta 2, and beta 3 for net 1, net 2, and net 3 respectively. And beta 1 is based on net 1, beta 2 is based on net 2, and beta 3 is based on net 3. To capture this importance bias importance, we further design a graph level attention to aggregate graph-specific representations. As a result, we obtain the final representation Y for microns and JAGs. With the representations for JAGs and microns, we can use them to reconstruct a micro-JAG bipartite network using a semi-supervised linear way. This matrix Q is a probability score matrix where each element represents the possibility of a micro pair to be associated. To evaluate the importance, to evaluate the effectiveness of our model, we compare our model with 7 state-of-the-art methods. We also use 3 different cross-validation settings to further validate the effectiveness of our model. Based on the evaluation, we can see that our proposed EJTMDA model performs the best under CVS 1 and CVS 3. For CVS 2, EJTMDA performs the best in terms of AUC. While the AUPR is relatively worse than that of BLM NII. Recall that we construct 3 networks to predict the micro-JAG association. And net 1, net 2, and then net 3. In particular, GraphNet is the network that integrates the 3 sub-networks. The results indicate that all of the 3 networks have to improve the prediction performance of our model. As net 1 is a real network, it performs the
1 minute left.
best.
Sorry.
It performs the best among 3 sub-networks. Net 2 contributes relatively more than net 3. While GraphNet integrates 3 sub-networks, its performance is undesirable due to the lack of graph-level attention. Overall, combining 3 sub-networks with attentions more benefits the prediction performance. In our proposed framework, we design a hierarchical attention mechanism to learn node representation, namely node level attention and graph level attention. The results indicate that both node level attention and graph level attention benefit the prediction performance of our model. And besides, node level attention is more important than graph level attention. Finally, we conduct the case study to further validate the effectiveness of our model. We take JAG simple flux casing as example. This table shows the top 20 predicted micros that are associated with JAG simple flux casing. The results indicate that 18 out of top 20 predicted micros could be validated by existing reports. For example, this is a paper published in 2018. The author confirmed that simple flux casing can generate activity against the Candida albicans. Sorry. This is all. Thank you.
So,
do you have a question?
Thank you very much, Yahui. We have time for 1 question. Is there any question? I don't see any then. Thank you very much. And then we can move directly to the last speaker of this first session, who is Gianmarco Messa, and he's going to tell us about the Siamese neural network model for the prioritization of metabolic disorders by integrating real and simulated data. So Gianmarco, you have 12 minutes.
Thank you. Good morning, everyone. I am Gianmarco Messa. I am a PhD student at King Abdullah University of Science and Technology. Today, I will talk about a project I've been working with Francesco Napolitano from the same institute in collaboration with Professor Elzea from Baylor College of Medicine in Houston and Professor Diego Di Bernardo from the Teleton Institute of Genetics and Medicine in Pozzuoli near Naples, supervised by Professor Shingao. So I'm going to talk to you about a technique we devised for the prioritization of metabolic disorders, in particular inborn error of metabolites that is based on machine learning, so Siamese neural networks, and integrates real and simulated data. In this presentation, I will give you a brief introduction on the problem, then I will talk about the methods we devised, then I will give you the results and our conclusions. So the problem at hand is a set of genetic diseases that involves the deactivation of one enzyme, or actually more, but we focused only on one, and interrupts the metabolic pathway. For example, we can see in the picture that in the urea cycle, the enzyme EC6.3.4.5 that catalyzes the reaction from citrulline to arginosuccinate can be deactivated. In this case, we have an accumulation of citrulline inside the cell and the bloodstream that will cause this kind of disease. It is actually quite easy to detect this disease once you know it. In fact, the citrulline is called a biomarker for this disease, and it is used for targeted screening on newborns. Our objective instead is to devise an agnostic method that is not aware of previous knowledge such as biomarkers, but uses untargeted screening, meaning the plasma sample is passed through a mass spectrometer and are generated metabolic profiles. These are usually analyzed with classical statistical methods. Instead, we decided to use a machine learning approach. So in previous works, most of the works are focused on targeted screenings, so the use of biomarkers there are also works that are focused on untargeted screening, meaning that they use classical statistical methods to find alterations in the metabolites to diagnose a metabolic disease. And also many studies are focused on the discovery of new biomarkers. We have been working with 3 data sets. 2 data sets are made of real data. They comprise a grand total of 168 metabolic profiles with more than 1,000 metabolites each. The classes of diseases, inborn error of metabolites, IEMs, are 21 because the second set has classes that are subset of the first one. And to make our method work, we used a metabolic network to produce 568 metabolic profiles using the knockout genes in the network. So after harmonization of the 3 data sets, that means matching the metabolites and removing the metabolic profiles that were not suitable for our study, we ended up with 124 metabolites per profile with only 149 profiles for 14 classes. So to produce our data, we use metabolic networks. These are a well-known framework that uses the flux balance analysis, which is a linear programming algorithm to compute the best possible fluxes inside the graph that is comprised of metabolites and reactions given an input and an output called objective functions. So we perturb this graph, this model, removing one reaction, and we can compute in this way the modifications that appear inside the various fluxes of the network, and in this way we can create simulated profiles for our metabolic disease. The metabolic networks are exclusively based on the stoichiometric metrics of the cell, and they do not take into account accumulation or any sort of ordinary differential equation. So since we didn't find any similar research through the literature, we devised the baseline model, so something simple we could relate to, that is just the L1 distance between the real and the simulated profiles. So once the distance is computed, we ranked the lowest by distance, the most to the least similar simulated profile to each of the real profiles, assigning then a label of the best simulated profile to the real profile. On top of this method, we devised a more advanced technique to take into account also the biases in the data and the fact that the sources of the two data sets are not exactly matching or perfect. So we built a Siamese neural network. This kind of neural network is focused on the fact that there is an encoding module that takes in input both the data sets indifferently. So it completely shares the weights, and there are not two encoding modules, even if the data sets may be slightly different from their sources, and then performs a distance and computes. Now, to validate our method, we decided to use two approaches. We used the standard k-fold cross-validation. In this case, we used a 6-fold cross-validation because it was the best possible solution without removing too many classes from our datasets, since some of them did not have enough number of samples to be validated using an higher number of folds. So we can see on the graph on the left that the Siamese neural network approach outperforms vastly the baseline model. Instead, to check for the ability of our model to learn about unseen diseases, we devised a zero-shot validation. This is an approach that falls into meta-learning, and it removes during testing, during training, and during testing, one class completely. So the model, instead of 14, sees only actually 12 classes, because one is used for validation, the other is used for testing. So from 12 classes, it is able to make a prediction about the missing classes, even if it has never seen them. This is much more similar to our baseline approach in the amount of knowledge we put into it. Going more into the details, we can see the Siamese neural network model in zero-shot validation analyzed disease by disease, and we can see that it performs very well on some diseases, but it doesn't perform so well on others, and mediocre in a mediocre way on others more. Why is that? We believed it was a problem of, for example, biomarkers. So we did the biomarker analysis to check whether they are fundamental or not on in our task. We can see that from top to bottom in this box plot, the biomarkers are very influent when using the baseline model. We see from the third box plot that with all metabolites, the baseline model is able to perform better than without biomarkers. Instead, if we remove random metabolites, we have a gain in our signal-noise ratio, so we get a better performance. Instead, the Siamese neural network is a more resilient method with respect to the information that it receives. So once we use data set without biomarkers, we can see that the performance drops, but not so severely with respect to the baseline model. And if we remove metabolites, we also lose some performance, which means that our Siamese neural network is able to rely on a wider variety of information that is present in the sample. In conclusion, we introduced an innovative approach using Siamese neural networks that integrates real and simulated metabolic data. This was our focus, to integrate real and simulated metabolic data, because gathering real data in such environment, all coming from the same studies, it's actually quite difficult. So we wanted to expand the possibilities of our research. Also, we wanted to aid the diagnostic process using untargeted metabolomic screening, because we believe it is the future for newborn screenings, instead of using the traditional ways that comprise several number of tests, one for each disease. In this way, it is possible to get all the information at once. And even if we need some improvements in our methods, we believe that with more data and with more metabolites especially, we will be able to get a grasp much more information from our data. This is a good step towards automatic diagnosis of metabolic disorders. And we hope to work on this project much more and include newer techniques, especially newer metabolic networks, and get results that could be reliable even for a medical application. Thank you very much. If you have any questions.
Okay, Gianmarco, thank you very much for the presentation. So I see a couple of questions here from Peter Bram Hohen. So first one is, are the real and simulated data available?
Yes, they are all available online. And once the paper will be published, there will be the link, but I can produce it now, actually. So give me a minute. I can link it in the... Actually, no, the data are available in the papers that are here, the real data. The simulated data aren't available. So Miller and Barrage are the two papers from which we took the real profiles.
And the simulated data?
The simulated data, we produce them using a Patonet, following the paper from, I don't remember the name of the paper, I'm sorry, but it will be, all the description of how to get the data will be in the paper. In more detail.
And would the matching of simulated to real data increase the chance of overfitting?
Yes, it does, actually. Since the network is very, the data are very few, the network had to be very small, not to avoid overfitting. And the fact that this kind of setup, it automatically tries not to overfit, because it tries to compute a distance, so it's a regression. And it doesn't learn by heart what is going on, otherwise it would be impossible to predict on other classes.
Okay, we have another question from Anna Niewes. Says, how do you address the non-quantitative nature of untargeted metabolic screenings in the simulated profiles?
Actually, we used quantitative data, and the simulated data are quantitative since they come from a simulation with actual values, and the real data are yet quantitative again, because we use the values in the profiles instead of the up or down that comes from the difference. So to compensate for this, we used actually the neural network was the idea to compensate for this, because otherwise it would be quite hard.
Okay, so thank you very much, guys, to all the speakers in this first session of the systems track. So we reconvene for the second session at 12:10. So after the keynote talk. So thank you very much again, and I very much enjoy your presentations. Ciao.